Hello and welcome to Medical Dialogues. I'm Anshika Mishra. Today we'll talk about gut microbiota influence, severity of respiratory viral infection. The composition of microbiota found in the gut influences how susceptible mice are to respiratory virus infections and the severity of these infections, according to researchers from the Center of Translational Antiviral Research in the Institute of Biomedical Sciences at Georgia State University. The findings, published in the journal Cell Host Microbe, report the segmented filamentous bacteria, a bacterial species found in the intestines, protected mice against influenza virus infection when these bacteria were either naturally acquired or administered. This protection against infection also applied to respiratory syncytial virus and severe acute respiratory syndrome coronavirus 2, which is the SARS-CoV-2, the virus that causes COVID-19. To maintain this protection, the study noted that segmented filamentous bacteria required immune cells in the lung called basally resident alveolar macrophages. In this study, the researchers investigated how differences in specific microbial species can impact outcomes of respiratory virus infection and how they might do so, which hasn't been well defined previously.
Are you also aspiring to excel in medical journalism and delve into the intricacies of health and medical reporting? Explore our comprehensive course on medical and health journalism available at the Medical Dialogues Academy. Learn from industry experts who possesses in-depth knowledge and experience in this field. For further details, scan the QR code provided or access the link in the description box below.
They studied mice with discrete microbiome differences and mice differing in the only presence or absence of segmented filamentous bacteria. Viral titers in the lung were measured several days after infection and varied significantly depending on the nature of the microbiome of the different animal groups. These findings uncover complex interactions that mechanistically link the intestinal microbiota with the functionality of the basal resident alveolar macrophages and severity of respiratory virus infection, said Dr. Andrew Gevritz, co-senior author of the study and Regents Professor in the Institute of Biomedical Sciences at Georgia State. The study found that in segmented filamentous bacteria, negative mice, basally resident alveolar macrophages, were quickly depleted as respiratory virus infection progressed. However, in segmented filamentous bacteria colonized mice, basally resident alveolar macrophages were altered to resist influenza virus infection, depletion, and inflammatory signaling. Thank you for watching. Stay tuned to Medical Dialogues for more updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.